This method can help answer key questions in the field of T-cell immunology and development of viral vector vaccines. The main advantage of this method is that only small amounts of blood are needed, which allows repeated measurements from the same mouse. Additionally, virus and transgene-specific CTLs can be detected from the same sample. Demonstrating the procedure will be Jasmin Rinofner, a technician, and Annika Rössler, a grad student. Measuring and analyzing the samples will be Zoltan Banki, a postdoc.
To begin, safely restrain a mouse according to the text protocol. Collect 20 microliters of blood in an EDTA-coated tube from the tail vein of a mouse. When working with splenocytes, isolate the spleen and use the plunger of a syringe to press the tissue through a cell strainer. After performing lysis on the erythrocytes, count the cells and adjust the concentration of the sample. Prepare and label one fax tube for each sample, and transfer 100 microliters of organ suspension or 20 microliters of blood to the tube. When working with splenocytes, centrifuge the organ suspension for 5 minutes and discard the supernatant. Then, vortex the tube to resuspend the cell pellet. For each channel to be used, also prepare a fax tube for a compensation sample. Prepare one additional sample as an unstained control.